about localized kinship and spatial genetic structure among reef patches of the eastern oyster. Okay, so to begin, I would like to address the issue of scale. Scale is a central measure of concern in ecology, and it is particularly important when estimating genetic structure or patterns in the genetic makeup of populations, because these estimates can vary depending on the spatial scale at which they are being examined. So, for example, two populations that are located a far distance apart can appear to have very few genetic differences. However, two populations over a local spatial scale or a small spatial scale can be significantly genetically different. And this is concerning because undetected genetic structure at the local scale can inflate estimates of migration rates and connectivity. So historically, there has been a long-held assumption that most marine populations are characterized by high levels of gene flow and connectivity. In other words, these populations have been assumed to be genetically homogeneous or well mixed because larvae have the ability to diffuse far and wide in the open ocean. It should therefore be unlikely for any two individuals to be related, or it is unlikely for any two individuals that settle within close proximity to be related. And the reasons for these assumptions are that it is very difficult to track larvae directly in the open ocean. Kinship analyses have emerged as this useful tool for examining the realized movement of individuals among populations. And recent advances in molecular markers are allowing researchers to discover that even in species with the potential for extensive larval dispersal, you can see significant levels of genetic structure and localized kinship. And these studies are really transforming our view of genetic connectivity in the open ocean. And just as a side note, pelagic larval duration, or the time that larvae spend dispersing in the ocean, is often poorly correlated with dispersal distance. And a striking example of this can be seen in the California spiny lobster, which displays a significant level of genetic structure all across its entire range, despite a very long pelagic larval duration of 240 days. So ecologists have long been appreciative of the effects of genetic variation on different population traits, such as performance and persistence. And rightly so, because genetic variation is the raw material for evolution by natural selection. And so genetic variation is imperative for an organism's ability to adapt in a changing environment. Genetic variation, especially in habitat-forming species, can determine the number and diversity of facilitated or associated species, and therefore has the ability to shape multiple levels of biological organization, from the individual to community structure and ecosystem processes. Relatedness and kinship analyses are a nice complement to analyses of genetic variation, and kinship can have a profound effect on ecological consequences as well, especially in benthic habitats that are generally characterized by high population densities, where individuals are usually sessile, and so they are interacting with their neighbors. And these interactions between neighbors in benthic settings are really dependent on the level of relatedness between the interactors. So these interactions can be competitive or cooperative, and it's really dependent on relatedness. And characterizing interspecific diversity is especially important in organisms that contribute largely to ecosystem function, such as oysters. And that brings me to my study system, Crassostrea virginica. It is an economically and ecologically valuable species. In addition to its harvest value, the eastern oyster provides essential ecosystem services such as sediment stability, improvement to water quality, and production of habitat to hundreds of other species that are also of economic and ecological importance. Unfortunately, due to habitat destruction and disease, only 15% of oyster reef habitat remains worldwide. And so any influences on these populations will be essential in the recovery and conservation of the species. However, genetic structure in C. virginica hasn't really been looked at these scales that are ecologically relevant. These studies have usually focused on the scale of tens to hundreds of kilometers, and that's where the aim of my study comes in, which is to characterize genetic structure in C. virginica over a local spatial scale to test the hypothesis that genetic homogeneity has really been overestimated in this species that has a pelagic larval duration of approximately 2 to 3 weeks. A secondary goal of this project was to investigate any potential relationships between intraspecific diversity and oyster size. So to test this, we went out and collected oysters from the research lease in Masonboro Sound, North Carolina. We collected oysters from 6 different reefs, comprising 3 different landscape or location types. So we collected oysters from 2 landward fringing reefs, 2 centrally located or isolated patch reefs, and 2 seaward fringing reefs. And they're just named seaward because of their position, which is closest to the Atlantic Ocean. And so we collected 60 individuals randomly from each reef patch and collected about 382 total. So first, we isolated the DNA from the gill tissue of each individual, and then we used PCR to amplify 22 highly polymorphic microsatellite markers, and we then separated the PCR products using capillary electrophoresis with a genetic analyzer. The genetic analyzer then produced an electropherogram, which allowed me to score each allele, and we did that for all 22 loci for all 382 individuals. It's good times. Chelsea helped me a lot. Thank you very much. And we used a program called Strand to do that. So just a note on microsatellites. Microsatellites are highly polymorphic sequences. They are simple sequence repeats that usually contain 1 to 5 base pairs that are repeated in tandem up to about 100 times. And the fact that they're highly polymorphic really helps us determine relatedness. They are also neutral, so any differences in genetic structure, any patterns in genetic structure that we see in the system should be attributable to gene flow. So we then uploaded these genotypes into different genetic software programs. We used GenePop, which allowed us to calculate genetic diversity indices such as heterozygosity and allelic diversity. We then used a program called Arlequin to run an analysis of molecular variance, which partitions the genetic variation within and among the different reefs. And we also used the same program to look at F statistics to calculate pairwise FST values, which give us an estimate of how genetically different the reefs are from one another. And we also calculated FIS, which gives us an indicator of the extent of inbreeding. We also used a program called Genodi, which calculates pairwise kinship coefficients. So this table just shows some basic genetic diversity indices. So for all of the sites, we saw that the mean number of alleles per locus was pretty high. So the microsatellites are polymorphic. They're doing their job. We also saw that there was a significant amount of inbreeding at each reef. And the observed heterozygosity was lower than expected at each reef, which matches up with the inbreeding coefficients. We also looked at the percent of loci that were in linkage disequilibrium. And linkage disequilibrium just means that two loci are showing a statistical association with each other that would be higher than expected if those loci were randomly segregating. However, when we looked at the patterns of the pairs that were in linkage disequilibrium with each other across the different reefs, those patterns didn't, or there was no pattern, I mean, their associations didn't hold up. So we can assume that they're not genetically linked, and each reef also was in Hardy-Weinberg equilibrium. So the microsatellites are doing their job and working as intended. So this first table shows pairwise geographic distances between the reefs. So for example, fringing reef 1 and central reef 2 are 112 meters away from each other. And then this table shows pairwise genetic distances for those FST values I was talking about. And this gives us an estimate of how genetically different the reefs are from one another. So the higher the number, the more genetically different two reefs are. And so when we compare FR2 and CR2, they are significantly genetically different. So the values in bold here are representative of significant differences. I wanted to run a Mantell test to test for isolation by distance to make sure that the reefs were not displaying these genetic distances simply based on increased geographic distances. So I ran the Mantell test and found that there was no significant relationship between geographic distance and genetic distance. So no isolation by distance is going on. And this is also apparent just looking at the values. So if we look at CR2 and FR2, these two reefs are close together. They're the closest together, 83 meters. However, they are significantly genetically different. If we look at the two reefs that are the farthest apart from one another, they are not genetically different. So no isolation by distance. This table shows the results from the AMOVA, or Analysis of Molecular Variance. We found that almost all of the genetic variation exists between individuals occupying the same reefs. However, we also found significant genetic differentiation between oysters residing on reefs of different location types. So the fringing landward reefs are significantly genetically different from those seaward located reefs and the centrally located reefs. And this was really surprising given that these reefs are only tens to hundreds of kilometers apart from one another. Meters apart. They're very close together. And this same relationship was also mirrored with different genetic software programs. And we wanted to look at what could potentially be causing these fine-scale genetic differences. So we turned to analyses of kinship. And kinship coefficients were calculated based on the probability of two alleles being identical by descent in two different individuals given the allele frequencies within the population. And we found that the mean kinship coefficients were significantly greater within reefs than between reefs. So an individual residing on a reef was more closely related to another individual on the same reef than to an individual on another reef. We also found that mean kinship coefficients were significantly greater within than among the different reef type locations. So individuals on the fringing reefs are more closely related to each other than when an individual is compared across the different reef types. And this really suggests that it is not simply local retention or limited dispersal of larvae that is occurring in this system. It instead suggests that different oyster families are occupying these different reefs and reef types. So we then wanted to look at the distribution of kin within and among the different reefs. So we binned individuals into kinship categories or specific levels of kinship based on their kinship coefficients, and we calculated the proportions of individuals belonging to each kinship category at each reef. We then created another data set where individuals were randomly shuffled among the reefs to give us a null distribution with which we could compare our observed proportions to. So this graph shows the proportions of siblings within each kinship category within each reef, and so it shows the difference between the observed and expected values. So for example, in Central Reef 1, we saw approximately 20% more quarter siblings than would be expected. And so in 5 out of the 6 reefs, we saw significantly greater proportions of quarter siblings than would be expected. We also saw that the proportion of half siblings was significantly greater in 3 of the 6 reefs. So I just want to take a moment to talk about the different mechanisms that could be producing these different patterns in localized structure and kinship. So one hypothesis is that kin return back to their natal reefs, and this supports what we are seeing here with more closely related oysters occupying the same reefs. So oyster larvae could leave the natal reef and return back to that reef of origin, or related individuals can leave their natal reefs of origin and travel through the water column as packets, avoiding diffusion and dispersal, or diffusion during dispersal, and settling back on the reefs. Or these oyster larvae could display preference for settling next to kin. There could also be interactions with the local environment between larvae and different settlement cues, such as light, salinity, and fine scale differences in hydrodynamics. And given that Mason Borough Sound is a very sheltered area in comparison to the Atlantic Ocean, low levels of water flow could lead to significant levels of larval retention. So another potential mechanism to think about is sweepstakes effects, where there exists an extremely skewed level of reproductive success within the population. So when environmental conditions are poor, and this is something that I find is really cool about eastern oysters, is that they can change their sex depending on the environment. So females, when conditions are really bad, when there aren't enough resources and she doesn't want to allocate all of those resources to producing expensive eggs, she can turn into a male oyster. And so if you have a lot of females in the population turning into males, leaving only a few females contributing to the next larval pool, then you can see high levels of relatedness within the population, simply based on the fact that the larvae only come from a few mothers. However, sweepstakes effects will only produce localized kinship of the patterns that we're seeing here if these siblings are staying together and settling together. Another intriguing mechanism is local adaptation. So differences in the intertidal zone can produce these strong environmental gradients where organisms are highly adapted to different microhabitats. So individual larvae could be traveling from one reef type to another and then not surviving on that different reef type. Okay, so another part of this project was to investigate any potential relationships between intraspecific diversity and oyster size, and we are not completely finished with this part of the project yet. It is an ongoing endeavor, But we're hoping to get more samples. So I looked at the differences in oyster size. I first ran an ANOVA and a Tukey test to look for any differences among these different reefs. And I found that the landward fringing reef oysters were significantly smaller than the oysters residing on the centrally located reefs and the seaward fringing reefs. I then wanted to run an individual regression for each one of these diversity metrics against oyster size, and two things popped out when I ran these regressions. So reef location type, which we already found was a significant predictor of oyster size, popped out, and then the proportion of kin at each reef showed a significant positive correlation with oyster size. And so this is the regression showing that as you increase the proportion of kin at each reef, the average oyster shell size also increases. And this is great, but I only have 6 points. So what did we do? We went to Amy Wilbur, and she gave us a bunch more samples. So she gave us 10 samples from the Pamlico Sound, which is in northern North Carolina, and we collected more samples from Helix Creek and Lockwood Folly. And just so you know, all of the samples that came from Pamlico Sound are from subtidal habitats, whereas all of the samples from here are intertidal. So when we try to parse out the differences from the subtidal oysters, or the differences in oyster reef type and location, these two factors are confounded because all of these are subtidal. So Dr. Kamel and I went out and we collected more oysters from Pamlico Sound, which are intertidal oysters, and we are going to add that to this data set once those are all up and running. I then decided to run an ANOVA and a Tukey test on the sizes of all the oysters from these 24 different populations, and found that the intertidal fringing reef oysters are significantly smaller than oysters that are inhabiting Pamlico Sound, all of those subtidal habitats up there. And the central oysters, or the centrally located intertidal oysters, display an intermediate phenotype. So I then ran a regression for all 24 populations and ran the mean kinship values for each reef against average oyster size, and found a significant positive relationship between the levels of kinship on each reef and the size of the oysters. So the higher the kinship values, the larger the oysters, and I controlled for reef type and still found that there was this significant positive correlation. And just some studies that are notable on C. virginica. In 2010, Knights and Walters did a study that looked at recruitment rates, and they found that when recruitment rates were either really high or really low in C. virginica, then population growth declined markedly. And they think that this is probably due to competition, and when mortality greatly exceeds the recruitment rate, then growth is going to be hampered. And when the recruitment rate is really high, then you get rapid aggregation and growth, leading to increased levels of competition between individuals, and that could also lead to mortality. So there could be competition and cooperation going on in these different habitats. And in 2016, actually just a few days ago, Hanley et al. Came out with a paper where they manipulated levels of relatedness in different cohorts of oysters, and then deployed these cohorts out into the environment